Good afternoon. Or
You want to introduce it? Okay, next, Dr. Vural Tagal is going to talk about modeling karyotype-driven adaptation to metabolic restriction, predict therapeutic response, and immunogenicity in cancers. I guess you work with Naomi, right? Did I right? Okay.
yeah, good afternoon, everyone. And thanks again to Sandy and Kristen and all of the other organizers just for this great second conference of the mathematical oncology. So today, as Mehdi mentioned, I will talk about karyotype-driven adaptations. And actually so I will give some examples how karyotype adaptations can lead to immunogenicity and also maybe some therapeutic vulnerabilities. So we work again on aneuploidy or chromosomal instability just because aneuploidy is a hallmark of cancer, as many of you might know. So what we mean about this is that the vast majority of our cells are diploid, meaning that we have only 2 copies of each chromosome. However, in tumors, now more than 90% of the tumors or solid tumors are aneuploid, meaning that they either have chromosome losses, amplifications, or in the extreme cases, as you can see in the last panel, whole genome doubling. So in the very right panel, you can see actually the complete profile of TCGA tumors about the aneuploidy. And as you can see across all cancer types, ploidy levels change very significantly. And here in the bottom, you can see also the whole genome doubling status, showing that aneuploidy, in fact, is so common and actually one of the maybe major vulnerabilities that we can target if we can create some better understanding. So for that reason, actually, in our lab, we wanted to work on aneuploidy in a hybrid system so that we try to first understand the cost and also benefits of aneuploidy biologically, and then model its evolution or dynamic evolution throughout the tumorigenesis and also therapeutic response patterns. So in this slide, you can see our research plan. Basically, very simply, we hypothesize that if the DNA content changes in cancers, then the demand for nucleotides and basic precursors of nucleotides like the carbon, phosphate, or oxygen sources like glucose, phosphate, or oxygen, or some of the vitamin or minerals are going to be in different need depending on the ploidy levels. For that reason, we performed long-term evolution experiments by testing multiple different nutrient deprivation conditions and check how karyotype changes occur in those cells. And in the second parallel research program, we also went one step further, and we hypothesized that if cancers have different needs to nucleotides, then maybe they would show differential response to the inhibitors of different enzymes in the nucleotide biosynthesis pathway as listed here. So we tested those inhibitors in our isogenic cell line pairs that have, again, similar genetic background, but different ploidies. So I'm going to show some of the data that we have. And in the first experiment, we tested whether phosphate, which is very important or essential component of nucleotide biosynthesis, we tested whether phosphate deprivation would cause any aneuploidy or ploidy change in cancers. And in one of the cell line representatives of gastric cancers, we tested the phosphate deprivation conditions. And here you can see the growth rates of 2 replicates we have in this experiment. And after about 3 to 4 months rate, with gradual decrease of phosphate concentrations, as you can see, growth rates decrease but recovered, as actually Mehdi also earlier showed some of the experiments similar design. We see that some of those cultures, even after dramatic decrease in the nucleotide phosphate deprivation conditions, they could recover and then restore their growth. Then we track those cells with very routine longitudinal microscopy images and analyzed also those images for both growth rate assessment and also some other morphological features. But at the end, we also tracked over time how their genome or ploidy changed. So on the right side, you can see the example. Actually, in this condition, we observed that phosphate deprivation didn't change any ploidy level in the cells, but caused a very significant ploidy shift from here. There are 3 to 4 different clones which seem to change over time. Towards the end of the experiment, the clone labeled or color-coded with orange starts to take over, and it is actually, it represents the gain of chromosomes 11, 18, and 21. As you can see here, in the control, those chromosomes are either lost or at very low hypo levels. However, in the phosphate-deprived conditions and the adapted culture, you can see there is an increase in those copy numbers. So then we wanted to ask what could be the consequences, the biological consequences of this ploidy shift over time under this phosphate deprivation. And since phosphate is one of the major metabolites, we performed the unbiased full metabolomic screen, and we basically measured 500 plus metabolites in these cells after adaptation. So we observed that the major change was in the tryptophan metabolism, and immediately we actually thought about its actually importance in one of the major pathways that is essential for NAD synthesis. So these pathways and the details are not that important, but here the one labeled in the middle, kynurenine, which is an important metabolite in this pathway. And actually, it is a well-known metabolite that has immunomodulatory role. A little bit like background about this is that kynurenine is, if it is present, it can actually inactivate the T cell maturation process. So here in our example experiment, you can see that NAD levels actually didn't change because cells under phosphate deprivation could utilize salvage pathway and still have NAD. However, they could almost completely or completely inactivated the kynurenine production. So maybe one important thing is that here is the NAD structure, and you can see 2 phosphates in NAD, which means that actually under this extreme condition, instead of synthesizing NAD de novo, they just switch to salvage pathway to use this phosphate for nucleotide biosynthesis. However, then we wanted to see if now these cells have some immunogenicity or increased immunogenicity, and we collected T cells from mice and collected also conditioned media from either control or phosphate-deprived conditioned cells or after the cells adapted. And we checked how T cells maturation occur. And as you can see here, the red indicates the increase in viability of the T cells, and CD69 positive, which is a marker that indicates the maturation of T cells increased in the condition or increased with the conditioned media coming from phosphate-deprived cells, whereas the normal cells didn't have the same effect. So overall, our results here show that maybe not genes actually by themselves, but the combination of genes can create a more complex biology. And then by understanding this biology, we can understand the evolution of tumors and maybe make better predictions like immunotherapy response or even the chemotherapy response. And in this case, our data suggests that karyotype landscapes can be biomarkers, and if some of those chromosome gains or losses are we might even categorize tumors like hot or cold tumors. So the second example is hypoxia. So under this condition, we used an isogenic pair of breast cancer cell line. And here you see the parental cell line with almost diploid DNA content. And then the isogenic pair has tetraploid content, meaning that almost every chromosome has 4 chromosomes, or 4 chromosomes for each of the other 23 pairs. So under hypoxic conditions, we wanted to test whether, again, any aneuploidy change occurs, because that is also one of the major characteristics of tumor microenvironment. And surprisingly, in this case, in the near diploid parental condition, we didn't see any significant change. However, we observed that this hypoxic environment couldn't tolerate high DNA content, and cells after about 2 to 3 months were selected against haploidy, and chromosome numbers went to almost near triploid level. So to see if this can happen actually in vivo, since hypoxia is one of the, again, very major characteristics of cancer, we wanted to test this in vivo in mice. And in actually mammary fat pad in mice, it is known that normal physiological oxygen level is 4% in the muscle. However, mammary fat pad is, especially in the young mice, 10-week-old young mice, it is about 2%. It has high environment. So we injected these cells, 2N and 4N tumors, into mice and mammary fat pad, and or to young mice, and actually, a little bit surprisingly, but also expectedly from the previous experiment, we observed that 2N tumors formed pretty rapidly with a median time of 15 days, whereas 4N tumors needed 42 days to form. And after the formation of these tumors, we collected them and dissociated these tumors to check the DNA content. And here on the right side, you can see flow cytometric results coming from the 4N tumors. And actually, we observed that the original 4N or near tetraploid tumors, when they injected, and then the tumors dissociated from these mice were all depolyploidized to 2N form, just confirming that there is a connection or a strong link between hypoxia and intolerance to haploidy. So the next thing, we wanted to also explore whether aneuploidy and this high profile can be targetable. And finally, we tested our 2N and 4N isogenic pair with a large drug set, and we identified that 18 drugs were cytotoxic or differentially cytotoxic, and they are listed here. Among those, I will show the results and modeling results with gemcitabine, because this was actually one of the top. On the left side, you can see that 2N and 4N cells under microscopy or under microscope, we observe that the 2N cells are hypersensitive to gemcitabine, whereas 4N cells could tolerate this treatment by changing to non-proliferative polyaneuploid giant cell state. And basically, we created 4 compartments just to model this interaction, and then we parameterize all of the values listed here, including the gemcitabine drug concentration, intracellular drug concentration with PK/PD data. And we use in this experiment linear chain trick-based model, because we had to check the phase durations of the different cell cycle phases, since it differs from cell, and that was just the best for data fitting. For interest of the time, I will briefly show the fitting data, and you can see here the different phases, and then transition to this non-proliferative state fit pretty well. And our goal is to use all this parameterization in the future to create a simulation for drug response, especially for the drugs that are differentially cytotoxic to either low or haploidy cancers, or according to karyotypes. So in total, or at the end, we identified that different microenvironment conditions, phosphate deprivation, hypoxia, and glucose deprivation specifically, create either ploidy shifts or ploidy changes. And there are a large drug or a good number of drugs that show differential cytotoxicity to either low or haploidy or in a ploidy-dependent manner. So in this project, I would like to thank all of the lab members and also our collaborators, especially Dr. Marusik. Here we use his resources a lot, and including Ana Gomez, Heather Hahn and Derek Duckett, Stuart Maudsley, and Dr. Bosler, Mark Davis. So we are now expanding this project, and we would be more than happy, or I would be more than happy to get more questions. Thank you very much.
Thank you. Perfect. Thank you, Vural. Any question for Vural? We have time. Andrew.
How generalizable do you think is this intolerance to hypoxia from polyploid cells? So that basically it leads to prediction that the combination of cytotoxic chemotherapies and VEGF inhibitors should be effective if it's generalizable. Is there any empirical evidence that this is the case?
We actually have a little bit complex observation. Just today, I wanted, for the interest of time, didn't want to mention that, yeah, there is a selection. Seems like there is a favorable karyotype, by the way, not like the cells have to go all the way to near diploid levels, but there is a favorable karyotype state around, in fact, near triploid state. So, but combining with some other drugs could be a next test we can test, but at the moment, yes, we don't have clear explanation for it.
Because you have fairly strong data that surviving chemotherapy requires this polyaneuploid state. So if this stage is really sensitive to hypoxia, it seems to be like a good opportunity to interfere, but is it generalizable?
Actually, it is true that polyploid giant cancer cell state is one of the resistance mechanisms for especially this drug. However, now we know that there are also multiple types of polyploid giant cancer cells, and they have different characteristics, including their tolerance or sensitivity to other insults like hypoxia. In this case, we tested this with gemcitabine and polyaneuploid cancer cell or polyploid giant cancer cells formed. And the hypoxia, the impact of hypoxia here was not checked yet, but that can be tested.
Hey, great talk and super cool results. I'm interested in the hypoxia as well, the loss of polyploidy after exposure to hypoxia. How about intermittent hypoxia? If you go back and forth, do you see any different evolution in dynamics or?
That is, in fact, one of the questions now in line, just because we have additional results, including radiation, actually, one of the tests we have done. We didn't talk about this, but maybe Dr. Neumann-Dor will talk about that part, and there we can go over the data more. So we observed that under hypoxia, at least at the last stage of this evolution experiments after adaptation, they actually become resistant to hypoxia as well, even if we put them into normoxia for a short period of time, like for a few days. However, now we wanted to test what happens if we put them in normoxia longer, because there are also other adaptations that require more time. For example, mitochondrial biology changes and so on. So they can come back, or whether that comes back or not. And more importantly, from our perspective, we are wondering whether ploidy would also come back, or at least we can now allow those cells just lose their advantage, the growth advantage. But yes, that's one of the next steps.
Perfect. Thank you so much. Thank you, Vural, again. So let's have a 20-minute break, and we all come back at 4:50, please, on time.